Hello, anatomy and physiology. Welcome to our first day of remote learning. We are going to just do a really fast one today to let you sort of get into the swing of things. My videos for the following days will probably be a bit longer because I'll be covering new content, but I sort of wanted to do a recap of what we've gone over so far for the students who've been in quarantine leading up to our remote learning 2 weeks. And if you haven't seen her before in a video, this is Penny. She loves to be in the videos, but she also snores like a monster. So if you notice a weird noise, it's her. So when we were in class last, we talked about what day our testing days were going to be. You A-Day students, it's going to be December 1st, B-Day December 2nd. A-Day students, this means that our review day will be that Tuesday before Thanksgiving. So if you want to knock your test out of the way early and not have to wait till after break to take it, you sure can. You just need to let me know by noon on Tuesday. I have another one joining me. Here's Norm. But you have to let me know by noon on Tuesday if you would like to take your test early, and I'll be sure to send you your testing instructions. I will only be sending tests remotely to students who ask for it. I won't do it for everybody, so you'll have to email me. If that's something you think you're interested in doing, just let me know by noon on Tuesday next week, not this Tuesday, but next Tuesday, if you want to take your test early. But if you just want to follow along with a regular pace, that's a-okay. And you guys can take your tests on the 1st or the 2nd in person when I see you after the break. Right. So these are the remote learning expectations I talked about with the students in class on Thursday and Friday last week. Make sure that you are checking in by noon on the attendance check. It's just a quick Google form, easy peasy to fill out. I have to put my attendance in by 1, so I need to know that you are here virtually by noon. And if you have any issues, text down, whatever, text me. That's fine too. I just can't keep track of a ton of texts, so having it in a spreadsheet is going to be keep me a little bit more sane and less likely to goof up your attendance. Plus, it's really easy for me to check what time you submitted it and what day you submitted it on. Please check Google Classroom. That's where these videos and any other things and assignments will be posted for you. If you've been in quarantine, please also use today, since it's a short day, to work on previous assignments. Use today to kind of get caught up with our current material as you would. And then make sure you're taking down your notes in your notes packet as you watch these videos. This is posted in Google Classroom, so you can print a copy from home. But I think almost all of you should have this by now because I gave you all of those things early for the, I was worried that something like this might happen and that we would be from home. And then work on your homework for the day it's assigned. So today you have an assignment, do it today. Tomorrow I will post it as being absent or missing, just so you know what you need to be working on in case you didn't get to something one day or another. And then we just went over the test days, but be sure that you get your Unit 10 material done on test day. All right. As always, let me know if you have any questions whatsoever. These notes we already took, and today's assignment is a review mostly of this, actually entirely of this picture here. I probably won't be on this slide long enough for you to write everything down, but this is talking about how blood cells form, right? And that all blood cells sort of start off as the stem cell called a hemocytoblast. And blood cell formation is this fun word to say, hematopoiesis, hematopoiesis. And then we have all of our different types of cells. Now, erythrocytes and thrombocytes are not leukocytes. This is a red blood cell, this is platelets. The rest of these, lymphocytes, monocytes, neutrophils, eosinophils, and basophils, those are all white blood cells or leukocytes. So when you're working on assignments, that differs and is important. Um, and in class on Thursday and Friday, we talked about the differences between these types of cells and how to identify them. So there's a video for you to watch. In addition to this, we'll talk about in a minute that will hit on some of these different types of cells. So our agranulocytes are these two cells, lymphocytes and monocytes down here, that do not have little dots or granules in them. And then we have the three up here, basophils, eosinophils, and neutrophils, that are granulocytes, meaning that they do have little, they're wind stained, their organelles sort of show up more speckly than the lymphocytes and monocytes. And then we went through the notes and talked about the different functions of each, which you'll see on the next slide in a wee bit more detail. And then we also have ones that eat things. That's a big main function of our leukocytes. Excuse me. Our leukocytes all have different functions, mostly to protect your body. 3 of them are eaters. Okay, our monocytes, because they're a macrophage, a big eater. Our neutrophils, those are phagocytic to bacteria, so they eat bacteria. And eosinophils, they are phagocytic to worms. They eat parasitic worms that might be in your blood. So those are our 3 eaters as far as our white blood cells are concerned. This image here was used to view the different types of cells on our microscopes. So that's also what we did on Thursday and Friday as we looked at blood on the microscopes, and your job was to identify the different types of tissues. This slide is within your student notes PowerPoint, so you can go back and look at this as long as you need to. It might be very helpful when you're identifying and working on that lab assignment that we did on Thursday and Friday last week. But as you can see, it's labeled here are granulocytes and agranulocytes. And then it gives you a little description under each as far as what you're looking for. So the neutrophils kind of have more of a palish pink stain to them, but they always have a multi-lobed nucleus. You kind of see like 3 little clumps here of the neutrophils. The eosinophils are a little bit more pink and they're bilobed, so just 2 little bumps to their nucleus. Basophils are super granulated, and so it's really hard to see through to the nucleus. Whereas lymphocytes and monocytes, they look more purple. Lymphocytes have a large spherical nucleus, and then our monocytes have more of a U-shaped or kidney bean shape to its nuclei shape as well. So then on Thursday, Friday, we listed out our most common to least common of the formed elements or of the cells. And I do want to point out erythrocytes, red blood cells, thrombocytes, platelets, and that's when our leukocyte list starts. So all of these guys down below are leukocytes. So neutrophils are your most common leukocyte. And again, here are some more notes that we took Thursday, Friday. Like I said, not a lot of new stuff for you guys today. We went through and talked about all of the different types of functions of each of these cells as well. And then our lymphocytes, I want to stress, they also fight your body, but from infection, but from a viral infection. Whereas a lot of those other ones listed up above here deal with bacteria or other invaders. But all of these guys, the TB and NK cells, they all fight viruses in your body. They're your immune response team, if you will. Okay, this is the one new thing here I just want to show you. This would have been our bell ringer had we had bell ringers to do today. So I mentioned this in our notes Thursday, Friday, but using this image, what type of cell produces thrombocytes? And excuse me. Yes, you see that there are those, the stem cells, but before it breaks down into platelets, which platelets I described as being cell fragments, it forms this thing called a megakaryocyte. So platelets are formed from megakaryocytes, and what happens here is in its final stages, this huge macro cell, a megakaryocyte, is broken down into small chunks, and that's where you get platelets from. So they're not like a real cell, but they're just like a large cell that was broken into clumps. And platelets we will learn about during hemostasis, which is blood clotting. They have a very important function. Okay, I'm not going to play this video here while I'm making my own video, but this is what I want you guys to watch today as far as new content goes. It is part 2, and we haven't watched part 1 yet, but it goes a little bit more in order with the cells that we are talking about right now. Since my video is short, I wanted to fill you in with a little supplemental video that has some animations and some material for you. Again, recap Thursday and Friday. This video is mostly meant for those students who've been absent because of quarantine. These notes are also in your student notes. We talked about different diseases relating to the blood, and some of the blood you see on the slides that we looked at in class for our blood smear lab are diseased. Not all of these diseases, I only have 4, but the 4 that are on there are on this list. So just super fast recap. Anemia means lacking blood, and there's different types of anemia. And then polycythemia is when you have excess blood. This is caused from blood doping, which you might be familiar with the Russians from the Olympics were accused of doing this, as well as um, Lance Armstrong, who won the Tour of France many times. Leukemia is cancer of your leukocytes, your white blood cells. So that's too many white blood cells. Infectious mononucleosis. This is a viral infection that attacks lymphocytes, and lymphocytes fight viruses. And this is why this virus in particular is really hard to beat. If you do get sick with it, it's long lasting, and you have fatigue for a very long time when you do end up contracting this virus. And then there are parasites that live in our blood. Malaria, it looks like it's caused from this creature called Plasmodium. It's a parasite, and it looks like little dots inside of your red blood cells, whereas trypanosoma, which is African sleeping sickness, that is a worm that lives in the plasma of your blood and eats the nutrients and good stuff that's in your blood. So it actually looks like a little worm inside there. So that was a brief overview of the different types of blood diseases, some of which you'll see on your lab. So this lab is posted within the Lab 01 assignment for Unit 10. So you can go through all of these different slides and get this information. So I had students write this information down in their notebooks. This is lab number 6, human blood smear lab. You at home can do it too, because I have pictures of everything you need. I gave you a hypothesis because formed elements, each formed element has unique functions. Its structure and quantity differ. This is noticeable when viewed with Wright's human blood smear slide. Is the hypothesis. So your idea was to observe the different types of formed elements and then fill out this observational data table, which is on this slide. So the idea here is that you would look at the sample, right, which I do have a picture of here, right, the one that's unlabeled. This kind of gives you, it's not circular, but it does give you a full view. You can use this as information for this data table. I want you to give a little sketch of the erythrocytes, just one. You don't have to do them all, right? Just one. The thrombocytes, they look like little specks or little dots. And then you would likely see a neutrophil, a lymphocyte, and a monocyte. Now, those three will not be in big numbers. And in fact, we have 5 leukocytes. The other two are not listed on there because they're in such small quantities. Then you're going to quantitatively estimate how many. This is a number. How many neutrophils are there? How many thrombocytes are there? How many erythrocytes are there? And erythrocytes are in large numbers, and so to count that will be very difficult. So I recommend taking that image and then maybe drawing on your screen if you can, take a screenshot of it. But you can cut it into quadrants, count how many erythrocytes are in a quadrant, and then multiply by how many that you have. Then I want you to make 2 observations and 1 inference for each formed element. An observation could be like, it's purple, it's red, it has little granules in it, just something that you observe with your eyes. An inference is an assumption about what it does. What do you think it does for your body, or its functions? Okay, and so you should be able to fill that table out, and then complete the lab assignment. This is just an image that helped us practice identifying our different cells, right? And you should see that as our, I think it's assignment 2. Again, this image here is used for those of you at home who can't actually view the tissue type. And then as a little bonus, I included images of abnormal blood as well. So these are a list of some of those diseases, not necessarily all of them that I just mentioned, but here's some pictures of the blood smears that were abnormal to see if you can figure out what's wrong with them. And this is in the form of a Google form for you to answer. And there are right and wrong answers for you for each of those different types to identify. And they go along with those diseases. So this video was mostly a recap of Thursday and Friday notes. Nothing new for those of you who were present. However, you do have a new homework assignment that does reinforce all of this information. Please watch the crash course video that's also attached, and then complete your homework and make up any homework you might be missing, especially for those of you who have been absent. Anyways, I look forward to seeing your name pop up virtually to know that you are at home working on your anatomy and physiology. Please email or message me on Remind if you have any questions, you get stuck on anything, or you're unsure of anything. Thank you so much. Have a wonderful day.